Okay, so I'm going to give you a quick tutorial on how to use the Scaffold software, which is what we're going to be using for the computer module for the proteomics data analysis. So the data that you're going to be looking at here is from a salmonella whole cell lysate. We wanted to reduce the complexity of this whole cell lysate, and so we've used one-dimensional SDS page gel electrophoresis in order to fractionate the sample by molecular weight. And so what we see here are some bands A through H that represent different molecular weight fractions of this whole cell lysate. A picture of this gel can be found in the PDF of your computer module assignment. And so what we're looking at right now is we're looking at the samples view of the Scaffold software. And we can see that there are multiple columns. This first column here represents all the proteins that were identified in all of the total samples that we've included here. We can see that that represents 625 different proteins with the current threshold settings that we have implemented. In addition to 625 protein identifications, we've also identified one decoy protein identification. And so if you remember with proteomics data analysis, I mentioned that we search our data against protein sequence databases, and then we also search them against a reverse or random version of that protein sequence database in order to allow us to calculate a false discovery rate and let us assess the threshold levels that we're using. So we currently have one decoy database hit that is represented above our current thresholds, and this represents a false discovery rate of 0.2%, and this is represented down here in this pink box. And so we can play with these protein identification thresholds by changing these probability thresholds in these drop-down options up here. Now, these represent identification probabilities that were calculated by the Scaffold program. And so this is all of the database search engines have different methods of reporting the statistical confidence and for setting thresholds. Scaffold will take all of these and convert them into a sort of universal format, and so it doesn't matter what database search engine you used. If you import the data into Scaffold, it will convert them to these probabilities. And so you can see that we can set these probabilities at both the protein level and also the peptide level. We currently have these set at 95%, which is a pretty safe place to be, but we can lower these. So if we lower this down to 20%, for example, we can see that we have significantly increased our number of identified proteins, but we have also significantly increased the number of matches to our decoy database. And our false discovery rate is now all the way up to 3.6%. So we'll go ahead and put that back to 95%. The other thing that we can change here is the minimum number of peptides. And so this refers to the minimum number of unique or different peptide sequences that we have to independently identify for a protein in order to be confident in that protein identification. So a minimum setting of 2 unique peptides is really the standard for reporting proteomics data in the literature. What this means is that we have to independently detect and identify 2 different unique peptides that match to the same protein sequence in order to say we have identified that protein with any confidence. Now we can change this, so we can put this down to 1, and we can see again that our number of total protein identification goes way up, but also so does our matches to the decoy database. So our false discovery rate has gone way up. So we're increasing our false positive detections, but we're also probably decreasing our false negative. The problem is we just don't know which is which. So the general rule is to keep this at 2 unless you're looking for something and you're trying to dig down to the data and look for something you know should be there and you just want to see some evidence that you might have detected it in your sample. We can also be even more conservative by moving this up to 3, 4, 5. Okay, So now we can see that we have a column for proteins. We have also a column for accession number and molecular weight of each of these proteins. Now these columns here represent the different bands from our gel, or the different fractions. So in a different type of experiment, these might represent replicates of your sample, biological replicates. It could represent different samples from different treatment conditions, and so on. So one of the really nice things about this software is that we're able to compare the results from different samples side by side. Now, as you've seen by my accidental clicking, all of these columns are sortable. So if we want to click and sort for column A, we just click on that column. We can also sort by protein name, so it will sort alphabetically, or by molecular weight, and it will sort by size. Now the values that are represented in these squares are dependent on what we have chosen in this display option drop-down. So right now we have it set to exclusive unique peptide count. So again, this refers to the number of different peptide sequences that we detected and identified for this protein in this sample. So in this case, for this particular protein, we detected and identified 40 unique peptide sequences, which all mapped back to this protein. And that is specific for this particular sample, sample A. So for this same protein, we detected only 6 and identified only 6 different peptide sequences in band B. So the last thing that I'll show you on this screen is this tab down here, or this part of the screen down here, which will let you link out to other external resources. And so whichever protein you might want to learn more about, you can click on the name here or highlight the protein in the list. And then we want to choose the appropriate database. And this data in particular was searched against a protein sequence database from UniProt. And so we'll want to select the UniProt database. And then if we click the accession number, this will link us out to the external UniProt database, where we can get a lot more information about this protein, including gene ontology. We can also do a protein blast search and get information about literature where this protein has been referenced, and so on. So within this software, we can also go to what's called the proteins view. So if we click on this tab, it will take us to a screen that looks like this. And this screen is a little bit more complicated, and we have these 3 different panels that are all sort of interconnected. So we start with this panel right here, and what this panel reflects is it's telling us which protein and which sample we're looking at. So we've got this protein here, and we can choose from any of the proteins on our list. And this is telling us that this particular protein, and we can expand out this box, was identified in all of our samples. And what this graphic shows us is it's a sort of graphical representation of the sequence coverage for this protein in this sample. So you can kind of quickly look and see that we detected a lot more peptides in sample A than we did for this protein than we did in some of the other samples. So whichever row we have highlighted in this quadrant, so right now we're highlighting this protein detected in band A, that corresponds to this list of peptides. So now what we're looking at over here is all the peptides that were identified for that protein in band A, the sample for band A. And down here, what we're looking at is the sequence coverage for that protein highlighted with all of the peptides that were detected for that protein in band A. And you can see if we choose the next one down, it will change in a minute. There we go. So now what we're looking at are the peptides that were detected for this protein in sample B, and here is the sequence coverage for that protein in sample B. And so again, the yellow corresponds to all the parts of the sequence that were represented by the peptides that were detected, and wherever you see a green highlight, that means that that particular amino acid was detected as modified. And so green M's mean oxidation of methionines. You may also see green C's, which correspond to that alkylating reagent that we use in the sample preparation step. Okay, so now we can see that we have this list of peptides, and one thing that you'll notice is that some of these peptides are listed more than once, and that's because we often detect spectra for peptides more than one time, because remember, the peptides aren't all entering the mass spectrometer at the same time. They're eluting from a chromatographic separation, and so they're entering the mass spectrometer over time that corresponds to a chromatographic peak. And we're using the data-dependent acquisition, which means that we're going to be detecting peptides more than once. So we can highlight a peptide here, and then if we go down and choose spectrum, we can actually visualize the experimental spectrum for this peptide. And remember that each of the peaks that's labeled as either a Y or B corresponds to a peak that matched the theoretical fragmentation for this particular peptide. And we can scroll down this list and start to look at all of the peptides for the different, or the spectra for the different peptides. And so, again, we don't typically look at all of these, especially when we've identified so many peptides for a given protein. But if we wanted to do some manual evaluation, then some of the things that we're going to be looking for are good coverage of the Y and B ions. So you can see here that we've got coverage of Y9, 8, 7, 6, 5, but we're missing Y4, but then we have Y3, Y2, and Y1. But they're pretty low abundance. But we have this corresponding B ions that give us a little bit more confidence in this identification. So we're looking for good coverage of the Y ions and the B ions. We're looking for good signal to noise. So signal to noise means that the signal from our peak is significantly above the noise level, and you can look at this and see the noise level. And then we're looking for the fact, we're looking at how many of the peaks that we detected are labeled or matched to the theoretical. And in this case, all of them did because they're all colored. But if we start to scroll down, we might see some examples like this, where we have some peaks that are black. And this represents peaks that we detected, but that did not match the theoretical fragmentation for this particular peptide. And so that might be an indication that that wasn't a great match. However, it doesn't really matter in the grand scheme of things if we're just deciding on whether or not we have confidently identified this protein because we found a lot of other peptides that have better matches. So the tutorial is going to take you through looking at some of these examples for multiple proteins. There's a whole bunch more information in this particular panel as well. So if we highlight any one of these, we can see that we have information on the actual mascot ion score. So mascot is the search engine that was used originally to do this, this search. These are the scores that mascot reports, and mascot sets its own threshold, which changes for every search, but in this search was 25. So what that means is that scores above 25, what mascot would consider good. And so we have a mixture. We have some that are above 20 or below 25 and some that are above. This delta ion score is actually very diagnostic. So this tells us what is the delta in the score for the next highest scoring match. So this is telling us that the high scoring match here was 24.2. The next highest scoring match was 15, lower than that. And so the higher this delta is, the better, the more confidence we have in our identification. The non-tryptic termini, number of tryptic termini was set to 2 in our data analysis threshold, so this should be 2 for everything. The modification tells us if we detected any modification. So again, this is going to primarily be either a methionine oxidation or that alkylation of our cysteine residues. We see some columns corresponding to our observed mass. So this is our observed m over z, or mass over charge, and then the actual mass, as well as the charge. And this information is represented down in the spectrum as well. So you can see that for this peptide, we observed m over z of 580, but this was a doubly charged ion, so what that means is that our mass was actually divided by 2 for the m over z. So the neutral mass is actually 1,159 Daltons. We can then see are the corresponding error. So this is the error of measurement in the intact peptide mass, and it's 0.62 parts per million. So this is a very high accuracy measurement. And you can see this here. They have a column for the delta mass, or the mass error, in Daltons as well as in parts per million. So the last thing I'll show you is this quantify tab. This quantify tab lets us do some preliminary sort of comparisons between samples. We're not going to focus on this quantitative scatter plots because we don't have a real quantitative experiment, or our gene ontology terms, which we didn't import for this data. But up here we can see these are just the total signal for this protein across all of our samples. And so this gives you some sort of, a very crude way of looking at sort of the relative abundance. So this particular protein was significantly more abundant in sample A than it was in some of the others. So you can really use this to get an idea about trends of abundance for a given protein across all the samples. We also can do we can do comparisons between up to 3 samples using this Venn diagram tool. And this is something we do quite often. And so right now we can see that it's set to compare band A, B, and C, and you can change these up as you want. But as it is, we can see that there were 43 proteins that were found in all 3 bands. And so if we're interested in those 43 proteins, we can see a list of them here, and we can export this list by right-clicking and saying copy all data and pasting that into Excel, or we can export it to Excel directly if we want, but I think it's easier just to copy and paste. We can also double-click on this, and it will apply this filter back to our samples list, and so now we're only seeing a subset, or 43 proteins out of 625, and then we can highlight these and link out to UniProt if we want to learn more, or we can also export the data from here if we go to Export Current View, and it will export everything on this page. So hopefully that will give you enough information to help navigate this software and be able to work your way through the proteomics computer tutorial.